Welcome to Signals and Synapses, the podcast of the Max Planck Institute for Biological Cybernetics in Tübingen, Germany. I'm Sophia Jans, and today I'm joined by Pascal Fries and Chiara Zanonato. Pascal runs the neurodynamics group here at the MPI for Biological Cybernetics. His research focuses on how the brain uses synchronized rhythms, for instance, when we pay attention or to control our posture and movements. Welcome to this episode, Pascal.
Hi, Sophia.
Chiara is a PhD student in the Dynamic Cognition group at our MPI. She will be co-hosting this episode and talk to Pascal about what the rhythms of our brain are and why they are so important for us. Thank you for joining us, Chiara.
Hi, Sophia. Hi, Pascal.
Many of our listeners will surely have heard of brain waves. For example, how beta and gamma waves are associated with dreaming and active thinking. Pascal, you mentioned in the pre-interview that you do not like the term waves here, actually, and you prefer brain rhythms, so we're going to stick with that. To kick things off, could you explain to us what these rhythms of the brain are?
Sure. So brain rhythms emerge when many neurons interact with each other. And very important is the fact that we have two key types of neurons, excitatory neurons and inhibitory neurons. Excitatory neurons activate other neurons, and inhibitory neurons suppress the activity of other neurons. And if you have these two forces, you have excitation that goes to other excitatory neurons, but also to the inhibitory neurons, and you have inhibition among the inhibitory neurons, but also back to the excitatory neurons, then you have these coupled opposing forces of excitation and inhibition, and that always leads to rhythms. And because many neurons participate in those rhythms, we talk about rhythmic synchronization or synchronized rhythms. And this is also why we can see these rhythms on the level, for example, of the electroencephalogram, the EEG, or the magnetoencephalogram, the MEG, or the local field potential, which is basically when you record something like the EEG on the brain or in the brain. You know? So because there are thousands or even hundreds of thousands or maybe millions of neurons that participate in these rhythms. And there are, of course, some rhythms that are quite local, like some gamma rhythm induced by a particular small stimulus inside the visual cortex. And there are other rhythms that encompass really large parts of the brain, like the alpha rhythm. When you close your eyes, there are probably, many millions of neurons participating in this alpha rhythm.
Is it even possible to have neuronal firing without brain rhythms?
That's a nice question. Yes, you first have firing, right? You said, can you have neuronal firing or neuronal activity, right? So I would say, well, that's the basic thing, right? You first have some activity, for example, some activity of some excitatory neurons. Mm-hmm. And as soon as that triggers inhibition and you get the feedback inhibition, then you start to engage in a rhythm. You can definitely have an absence of some particular rhythm. So, for example, as Sophia already mentioned, right, so there are different rhythms that are characteristically occurring in different frequency bands. We already named some of them, like the famous alpha rhythm that often occurs around 10 hertz, the one that is very strong and the first one probably to be described historically. And then there's the beta rhythm at about 20 hertz and the gamma rhythm, which is often occurring between 40 and 90 hertz. And it's definitely possible that at a given brain region, in a given situation, one of them or two of them is not there, and another one of them is dominant. But whether there can be a state without any rhythm is a super interesting question. I hardly ever thought about this. I would say that probably in a normal, physiological, non-pathological brain, in an awake state, you would probably always have some rhythm around.
Okay, and here there is a point that actually I'm curious about. So if brain rhythms naturally occur in any group of neurons, one challenge is providing they have a functional role, right? So let me ask you this way. Do brain rhythms always have a role?
Super interesting question, right? And this is exactly actually what has been the core question among our research for many years, right? And we've taken it basically a little bit the other way around. So our hypothesis is that rhythms just emerged out of kind of the necessity that I mentioned earlier, right? So there obviously is excitation, so neurons excite other neurons, and there needs to be inhibition to keep excitation in check. So there is inhibition back to the excitatory neurons, and then this emerges. This rhythmic interplay of excitation and inhibition emerges, and to the best of our knowledge, all the physiological rhythms. So I'm not talking about epilepsy, for example, but all the physiological rhythms are what we call inhibitory rhythms. So rhythms where the feedback inhibition plays an important role in generating them. These rhythms. And then we ask the question, okay, if these rhythms are anyhow there, could they possibly not influence processing, right? You asked, do all rhythms have a function, if I remember correctly, right? And we think, okay, if these rhythms just emerged, could they possibly not have a consequence? And our working hypothesis is that this is not possible, right? That a rhythm, if present, is a emerging population phenomenon that involves thousands and millions of neurons, and it temporally structures their activity. So we cannot really imagine, or let's say the work, scientifically speaking, right? Our working hypothesis is that this must have a proximal consequence on the processing of those neurons. And then our extended working hypothesis is that teleologically speaking, basically evolution has used these consequences and has basically figured out that this could be good for some function, right?
Okay, super interesting. I would move on to the specific frequency we already started to talk about. Actually, I have a question about why we have, in the first place, these different brain rhythms. And I think you already mentioned, but I want you to explain me a little bit better this point. So, are these different brain rhythms a matter of the brain being in a certain state, or is it because of the properties of the specific neural ensemble firing, or it's a mixture of the two?
Yeah, again, super interesting question. So maybe I start by saying that the different brain rhythms are clearly associated with different states or different conditions. So, for example, I mentioned the alpha rhythm. And the alpha rhythm has been found when participants of an experiment closed their eyes. And they were just in an awake but relaxed state with the eyes closed. Now, there is another rhythm that is in a similar frequency band, also close to 10 hertz, but that emerges in the area that is we call the motor strip. So it's basically primary motor cortex and primary somatosensori cortex. And that's called the mu rhythm. It has a very similar frequency, as I mentioned, around 10 hertz. Also there, it is a rhythm that is strong when the motor system is not engaged. But for example, when you then actually move, this becomes weaker and a rhythm in the gamma frequency band starts. That's strong when you move, and it's strong also when you hold a strong force. And for example, in the visual system, that same rhythm, a gamma rhythm, is strong when a visual stimulus is presented. Kind of on a very general scale, one could say that the gamma rhythm is a rhythm that emerges when a local group of neurons is like functionally activated. When visual cortex is processing a visual stimulus, you see gamma. When motor cortex is actually doing a movement or holding a strong force up, then you see gamma. When the motor system is kind of awake but not doing a movement, you see much more of this alpha. When the visual system, the eyes are closed and it's basically in in a state of relative inactivity, you see a lot of this alpha band activity.
I see. So the same area can actually have different brain rhythms at the same time?
Absolutely.
Okay.
Yeah, I mean, primarily at different times. So this alpha gamma is a bit of there's a tendency of either or, right? So the more you get gamma, typically the more alpha goes away, but definitely they also coexist, right? This is not a black-white thing.
So, I mean, nowadays you mentioned we have EEG to measure this, but how did people discover brain rhythms in the first place?
I think EEG was the first method that discovered brain rhythms. As I mentioned, to the best of my knowledge, the alpha rhythm that was measured by Hans Berger with an EEG system was the first signal that was ever recorded from the human brain, to the best of my knowledge.
What did people make of it at the time?
Again, I'm not an expert in this, but my understanding is that Hans Berger was actually interested in phenomena that today we would probably actually call esoteric, right? In telepathy. Again, I didn't study this in detail, but my understanding is that he basically had two persons sitting back to back, basically, not seeing each other, not talking to each other. And he was recording EEG from their heads, from their scalps, and wanted to see whether there is something going on between them, some synchronization, some correlation or something like this. And when they closed their eyes, he just realized, oh, wow, a strong rhythm emerges. And that was the alpha rhythm. And then, of course, many studies started, right? So this is often the case in science, right? Science is often driven also by methods and by the availability of a signal, of a window onto the brain, right? And suddenly there was a window, if you like, on the human brain. And of course, many people got interested in this on the one hand from the medical side, right? So there were then many studies investigating this alpha rhythm with EEG in different medical conditions, in different psychiatric conditions and neurological decisions and conditions and neuropsychological conditions, so after brain lesions. And there was also a lot of interest from the side of the psychologists who suddenly said, oh, fantastic, right? We can not only study the brain by asking questions or asking people to push a button, but we can directly measure from the brain. And of course, they also then did a lot, But for many decades, I would say, a lot of this was focused on basically an approach that I would call psychophysiological. So you would create a psychological condition, for example, being alert versus being not alert, being attentive, not attentive, memorizing versus nothing in memory or something like this. And then you would find a correlate. You would see that alpha was stronger, weaker, or whatever, right? And something that in my perception came somewhat later was the question, okay, can we come up with a plausible hypothesis about how such a rhythm would actually contribute to the function, right? Not just a correlation, but can we really make the case, okay, because there is a rhythm, this and this happens in the brain, and therefore this supports, for example, attention. And this is exactly what we have been trying to do with our work, to say, because this rhythm synchronizes with some other brain areas, this information is going through, and that's attention, for example, that supports attention.
I think we're at a super interesting point here because, this is, Chiara, what you told me before, where you started getting into Pascal's work when you were still an undergrad student, right?
Exactly. I have to say I'm super excited because this working hypothesis of communication through coherence, which is your super famous paper of 2005, is the way I got to know you. So I'm super excited to be here. But I would like to slowly unpack this whole idea. So I think I would start by asking you what was the mainstream idea before your paper about the communication between neurons.
Well, I would say that basically the mainstream idea did not include rhythms, right? So the mainstream idea is that we have different layers in the brain. And at each layer, you have many neurons horizontally, right? And they contain information. So for example, in visual cortex, in primary visual cortex, this layer of neurons would somehow represent the outside visual world that comes in through your eyes, right? You can envisage it a little bit like an image, but already a little bit processed, okay. And then this goes forward to the next layer. And in this going forward, there is some processing happening, and basically some sort of filters are implemented that find out whether in this input there is a particular structure, whether there is, in the end, whether there's something interesting like a face or so. So there is a sequence of filters that find out whether there is, for example, a face or maybe some food or maybe some other relevant thing, okay? And rhythms didn't play a role.
Okay. And then you suggested an hypothesis that actually implement the rhythms into this whole processing. Can you give us a gist of it?
Yeah, as you already said, it will be a bit of a challenge for us to unpack that for the listener, right? And let's try. So maybe I would say there are two things coming together, right? So on the one hand, these ideas about how the brain filters inputs and finds out that there is, for example, a face in the input. And on the other hand, simply the physiological observation that there are these rhythms. And we started with our thinking from the physiological observation. Maybe also because I'm a medical doctor by training, this for me was simply a super interesting phenomenon that was the basis for our thinking. And a key aspect about these rhythms that I mentioned already a few times is that it's a sequence of excitation and inhibition. And we simply reason that if a neuronal population is undergoing a sequence of excitation and inhibition, then during the inhibitory parts, the inhibitory phases, as we say, of this rhythmic cycle, these neurons will be less susceptible to input. So there will be basically phases where they are very susceptible to input, when they are anyhow in an excited phase, and phases where they are less susceptible to input, when they are in this inhibitory phase. And then we reasoned, okay, if these neurons now get inputs, these inputs will be effective during some phase and not effective during other phases. And we realized that this is a super interesting way of changing what is called input gain. This is a very central concept in brain research, what's called gain. You can imagine that, neurons are connected to each other, and one that's called a synapse, anatomically it's called a synapse. And this synapse has more or less a fixed strength, so it has a fixed impact on another neuron. And now, by having this rhythmic inhibition, the actual effective strength of this input is basically modulated. Or in other words, the gain of it is regulated. It's like you have a volume knob that is turned on and off all the time in a rhythmic way, right? And so then we realized, okay, hang on. When inputs would always come in when the knob is turned up, they would be effective by going through. And when they would come in at random times, they would be relatively ineffective.
So you were reasoning about the input, not really the state of the neuron, because the state of the neuron is in itself rhythmic, right? Like a neuron, as you said, cannot be always like in an excitatory phase. So you were more interested about the way the input is provided, not about the neuron itself.
Yeah, indeed. We were thinking, at this moment, about a group of neurons in a higher area, right? For example,
I think you need to explain to our listeners what higher area means.
Exactly. So in the visual system in the brain, the visual input comes in what we call the primary visual cortex, and then there are many layers, many areas in which there is more and more complicated processing. Basically, more and more complex features are detected. So in the primary visual cortex, for example, neurons respond to spots of light or line segments, these kind of things. And then in the next layer, they respond to simple patterns, spirals, gratings, stripe patterns. And then at one point, you get neurons that are selective for very complex things like, for example, faces or objects like cars, houses, trees, plants, whatever.
So when you say they're selective for, you mean they start firing when that's what their input, when that's what they get from the input.
Exactly, yeah.
Right. But back to communication through coherence.
So back to communication. You asked me, did we think about the rhythm from the perspective of the input or so? And indeed, so we basically said, okay, at a higher level, let's say even such a patch of neurons that would be selective for a relatively complex object, in a higher area, they also engage in these rhythms. And when they engage in these rhythms, they are only input susceptible for input that comes in at their own excitatory phases, whereas input that would come in at other phases is not so effective. And then we reasoned, okay, so these neurons, they get convergent input. And often this convergent input contains two different objects. So for example, there might be a face, and next to the face there might be a tree.
Right.
And when you pay attention to the tree, the face-selective neuron is not getting very active. That's a finding that had been well established. And when you then direct your attention to the face, this face-selective neuron gets very active. This work had been done by other labs, and it had been argued that this can be explained by the inputs representing the face being modulated in their input gain. And that's what we talked about a minute ago, right? So when you direct attention to the face, it seems as if the gain of those inputs that represent the face is upregulated. And then we reasoned, hey, didn't we just think that the rhythms rhythmically modulate the gain? So don't we know that these inputs, where they come from, they are also rhythmically active, right? So these neurons in earlier visual areas where the face is represented in primary visual cortex, they also engage in this rhythm. So the only thing that these neurons in primary visual cortex, where the face is represented and where it's rhythmically active, the only thing they would need to do is they would need to synchronize the neurons in the higher areas, and thereby they would automatically put their input always in at the right phase, right? And the other neurons that would represent the tree, they would come in at random times because they would not in that moment synchronize to the higher areas. They would still be rhythmically active, but they wouldn't manage to synchronize to the higher areas. So their inputs would come in at random phases, and they would often hit these inhibitory phases, and thereby they would just be blocked out. And so this fascinated us. And this is basically exactly what we called, you mentioned it already, the communication through coherence hypothesis.
I think I have a question about this synchronization. So is attention that is making this happen? So like how does synchronization work?
Again, you are asking super questions. So I think it's very important to dissect it, right? I don't think that attention makes the rhythms to begin with, right? So you can see the gamma rhythm even in an anesthetized individual, right? If it's an appropriate anesthesia, there are also anesthesias that kill the gamma rhythm, but under an appropriate anesthesia, you can see this when attention is almost certainly not there, right? So the gamma rhythm in primary visual cortex is not generated by attention. It's generated by a very, very basic physiological mechanism. There's simply the visual stimulus activates these neurons. There's excitation, inhibition, the interplay between excitation and inhibition. You have the gamma rhythm, right? The question is, what is then the role for attention, right? And based on our work, we believe the following. So when you open your eyes and there are, let's say, 10 different objects in front of you. 3 faces, 7 trees, let's say, to stay in this picture, right? Like there are 3 people in front of you standing in front of a few trees. So then you would actually find 10 different local gamma rhythms in your primary visual cortex. Each of those, let's say, objects is inducing a separate gamma rhythm locally. And these different gamma rhythms are not synchronized to each other. And it seems attention is then enhancing the frequency of the attended gamma rhythm. And the enhanced frequency is helping this, let's say, attended gamma rhythm to do the entrainment and synchronize the higher areas.
Okay, super interesting. I guess that my question now is about other possible ways of synchronizing or communicating. So we have just seen now that the synchronization is happening from the lower layers to the higher layers of the visual system. But I'm wondering whether the other way around is also possible. So from higher layers to the lower layers.
Absolutely. And this is, again, a super interesting question. So indeed, as we just said, so from the lower layers to the higher layers, what we call bottom-up communication, the gamma rhythm is very important. And we know that from the higher layers to the lower layers, so in the top-down direction, there are as many anatomical connections. So of course we wondered whether there's also gamma band synchronization going in this top-down direction. And interestingly, when we investigated that, we found that the communication in the top-down direction is much less happening through the gamma rhythm and is much more happening through the beta rhythm. And this seems to be a principle that holds at least across the visual system.
Super interesting, super interesting. So I think that we are close to the end of our podcast. Before saying bye to each other, I would like to know what are you currently working on here at Max Planck? What is your interest right now?
At the moment, we are very interested in an observation that we actually made serendipitously and published it in the year 2014. So for these studies that we talked about, we often record many trials, right? So in order to have strong data, we don't present a visual stimulus one time, but maybe 500 times. And one of my postdocs, he just looked at these data and he came to me and said, I can't believe it. This gamma rhythm is getting stronger and stronger and stronger over time. And at the end of 500 trials, it was 3 times as strong as at the beginning. And we thought, this is impossible. Like we looked at this rhythm for years before and investigated, hadn't seen this phenomenon, right? So we got super interested in this and basically developed the hypothesis that this is also important for some form of learning about the stimuli. And there had been another finding that had been described many, many times, which is a bit counterintuitive. When you record just the activity level of neurons, which is often taken to be the number of action potentials they fire per second, then when you repeat a stimulus, this activity level is actually going down, which is called repetition suppression.
Right.
I mean, it's very commonly observed, which seems to be a bit curious because it's not like you cease to see a stimulus, right? If a stimulus is repeated 100 times, it's not like the stimulus goes away for you, but the neurons start firing less. And we found that, okay, the neurons fire less spikes, but those spikes seem to be better synchronized in the gamma rhythm. And so we thought maybe these neurons maintain their ability to communicate the information about the stimulus to higher areas by firing fewer spikes, but those spikes more synchronously, and thereby entraining the neurons better and communicating their message more effectively, basically. And then we started studying this thing more and more and more. And so recently we found out sometimes it's also super interesting to use these serendipitous findings, right? So far we talked a lot about work that has been really hypothesis-driven, which is often taken to be the high road of science, right? But I also like it very much if you have a serendipitous finding and you just drill deeper and you find interesting things and you drill even deeper and it's getting more and more interesting. And this is one thing that at the moment fascinates us a lot.
And that brings us to the end of today's episode of Signals and Synapses. A big thank you to Pascal Fries for sharing his insights and expertise, and to Chiara Zanonato for co-hosting and guiding the discussion. It's been a real pleasure having you both on the show. And of course, we're grateful to all of you for tuning in. As always, you can find additional resources in the show notes. If you enjoyed today's conversation, don't forget to subscribe and spread the word. Until next time, stay curious and keep listening.